Gossypium is the cotton genus. It belongs to the tribe Gossypieae in the mallow family, Malvaceae, native to the tropical and subtropical regions from both the Old and New World. The genus Gossypium comprises around 50 species, making it the largest in species number in the tribe Gossypieae. New species continue to be discovered. The name of the genus is derived from the Arabic word gauze, which refers to a soft substance. Cotton is the primary natural fiber used by modern humans. Cultivated cotton is also a major oilseed crop, as well as a main protein source for animal feed. Cotton plants thus have an enormous weight in the world economy and are of great importance for the agriculture, industry and trade of many tropical and subtropical countries in Africa, South America and Asia. Consequently, the genus Gossypium has long attracted the attention of scientists. The origin of the genus Gossypium is dated to around 5–10 million years ago. Gossypium species are distributed in arid to semi-arid regions of the tropics and subtropics. Generally shrubs or shrub-like plants. The species of this genus are extraordinarily diverse in morphology and adaptation, ranging from fire-adapted, herbaceous perennials in Australia to trees in Mexico. Cultivated cottons are perennial shrubs, most often grown as annuals. Plants are 1 to 2 meters high in modern cropping systems, sometimes higher in traditional multi-annual cropping systems, now largely disappearing. The leaves are broad and lobed, with 3 to 5 lobes. The seeds are contained in a capsule called a bulb, each seed surrounded by fibers of 2 types. These fibers are the more commercially interesting part of the plant, and they are separated from the seed by a process called ginning. At the first ginning, the longer fibers, called staples, are removed, and these are twisted together to form yarn for making thread and weaving into high-quality textiles. At the second ginning, the shorter fibers, called linters, are removed, and these are woven into lower-quality textiles. Commercial species of cotton plant are G. hirsutum, G. barbadense, G. arboreum and G. herbaceum. Many varieties of cotton have been developed by selective breeding and hybridization of these species. Experiments are ongoing to crossbreed various desirable traits of wild cotton species into the principal commercial species, such as resistance to insects and diseases, and drought tolerance. Cotton fibers occur naturally in colors of white, brown, green, and some mixing of these. Most wild cottons are diploid, but a group of 5 species from America and Pacific Islands are tetraploid, apparently due to a single hybridization event around 1.5 to 2 million years ago. The tetraploid species are G. hirsutum, G. tomentosum, G. musclinum, G. barbadense, and G. darwini. Selected species. Subgenus Gossypium, Gossypium arboreum L. tree cotton, Gossypium herbaceum L. levant cotton. Subgenus Houngenia, Gossypium raymondi ulbr. One of the putative progenitor species of tetraploid cotton, alongside G. arboreum, Gossypium thurberitod, Arizona wild cotton. Subgenus Carpus, Gossypium barbadense L. creole cotton, Gossypium darwini G. Watt Darwin's cotton, Gossypium hirsutum L. upland cotton, Gossypium musclinum Mayers X. G. Watt, Gossypium tomentosum nut X. seam Mao Hawaiian cotton. Subgenus Sturgia, Gossypium austral F. mule, Gossypium sturtionem J. H. Willis, Sturt's desert rose. Formerly placed in genus Gossypium, Gossypioides brevilinatum J. B. Hutched, Gossypioides kirki J. B. Hutched, Cochia drinario edis lutin. Gossypium genome. A public genome sequencing effort of cotton was initiated in 2007 by a consortium of public researchers. They agreed on a strategy to sequence the genome of cultivated allotetraploid cotton. Allotetraploid means that the genomes of these cotton species comprise two distinct subgenomes, referred to as the It and Dt. The strategy is to sequence first the D genome relative of allotetraploid cottons, Raymondi, a wild South American cotton species because of its smaller size due essentially to less repetitive DNA. It has nearly one-third the number of bases of tetraploid cotton, and each chromosome is only present once. The A genome of G. arboreum, the Old World cotton species, would be sequenced next. Its genome is roughly twice the size of G. Raymondi's. Once both A and D genome sequences are assembled, then research could begin to sequence the actual genomes of tetraploid cultivated cotton varieties. This strategy is out of necessity. If one were to sequence the tetraploid genome without model diploid genomes, the euchromatic DNA sequences of the AD genomes would co-assemble and the repetitive elements of AD genomes would assemble independently into A and D sequences, respectively. Then there would be no way to untangle the mess of AD sequences without comparing them to their diploid counterparts. The public sector effort continues with the goal to create a high-quality draft genome sequence from reads generated by all sources. The public sector effort has generated Sanger reads of BACs, phosmids, and plasmids, as well as 454 reads. These later types of reads will be instrumental in assembling an initial draft of the D genome. In 2010, two companies completed enough Illumina sequencing to cover the D genome of G. Raymondi about 50x. They announced they would donate the raw reads to the public. This public relations effort gave them some recognition for sequencing the cotton genome. Once the D genome is assembled from all of this raw material, it will undoubtedly assist in the assembly of the AD genomes of cultivated varieties of cotton, but a lot of hard work remains. Cotton pests and diseases. Pests: boll weevil Antinomus grandis, cotton aphid Aphis Gossypieae, cotton stainer Distachus kernigii, cotton bollworm Helicoverpazia, and native budworm Helicoverpa punctigera are caterpillars that damage cotton crops. Some other Lepidoptera larvae also feed on cotton. See list of Lepidoptera that feed on cotton plants. Green myrid of sucking insect. Spider mites Tetranicus urtici, T. ludeni and T. lambi. Thrips Tabaci and Franklin yellow Schultze. Diseases: Alternaria leaf spot caused by Alternaria macrospora and Alternaria alternata. Anthracnose bell rot caused by Coletotrichum Gossypieae. Black root rot caused by the fungus Thelaviopsis basicola. Blight caused by Xanthomnus campestris PV. Malvaceae. Fusarium bell rot caused by Fusarium spp. Phytophthora bell rot caused by Phytophthora nicotianae var. Parasitica. Sclerotinia bell rot caused by the fungus Sclerotinia sclerotiorum. Stigmatomycosis caused by the fungi Ashbia Gossypieae, Erymothecium corally, and Doreobasidium pullulans. Gallery.